This is the first part of your DNA notes. So let's start with structure. DNA stands for deoxyribonucleic acid. We'll walk through that in a minute. All your DNA exists as chromosomes. Chromosomes are made up of DNA wrapped around proteins called histones. I'll show you a picture of that in a second as well. If you are a eukaryotic cell, which means you have a nucleus in all of your membrane-bound organelles, or maybe not all of them, but you have some at least. Your DNA exists in the nucleus, and it's trapped in the nucleus unless you're doing mitosis, in which case the nucleus kind of breaks down to get out of the way. And then each strand of DNA becomes one chromosome. If you're a bacterial cell, remember you don't have a nucleus, so your DNA isn't there, but you still have DNA. The only difference here is that the DNA is a circular molecule, and bacteria only have one of those, so only one strand of DNA and only one chromosome. So let's take a look here real quick. Here's your little bacteria. Let's just go with it. It's a little E. coli bacteria. You can't really see it in here because I don't have it, but they would also be ribosomes in the bacterial cell. But you would not see any other really complicated structures. Remember, there's no nucleus and no membrane-bound organelles. Here is the single chromosome. Notice that even though it's all kind of looped up on itself, it is just one chromosome. Notice that it's also circular, right? If you stretched it all out, it would be a circle. The fancy name for that is a plasmid. You'll need to know this later on in the unit. A plasmid is the name of that circular chromosome that bacteria have. But if you zoom in to its plasmid, or its strand of DNA, you'll notice that it's structured the exact same as it is in eukaryotes. You have the typical C, A, G's, and T's that you're probably familiar with from middle school. That does not change because it's a bacteria. It's just a circular chromosome rather than a linear one. So in eukaryotes, here's what you're probably a little more familiar with, your typical, in this case, double-stranded chromosome. So this chromosome has already copied itself. So if you want to throw some more vocab in, that would be a sister chromatid. Right, as would the one right next to it. Remember, this is the chromatid here. And then that is the chromatid. It's very tempting to want to do it like that, like the letter X. Just a pet peeve of mine, but whatever. Remember, right in the middle is the centromere. But nonetheless, these are linear chromosomes, and that's a difference with bacteria. These chromosomes are supercoiled. I'm just kind of showing you the structure here. These coils are made up of nucleosomes. This is not something you really have to pay too much attention to. And then if you break down a nucleosome, you'll notice that the actual DNA, this blue strand, is wrapped around histone proteins. And one of the reasons this works, or why this forms the structure it does, is because histone proteins have a positive charge. These little purple balls are the histone proteins. And the DNA is actually negatively charged. So DNA, right up here, DNA is negatively charged, and it wraps around the positively charged histones. That's how it's able to condense into that visible chromosome that you're probably a little bit more familiar with looking at, right? But at the molecular level, when you get all down to it, DNA, is a double helix, which we'll talk about, and it has the A's, the T's, the C's, and the G's, just like the bacterial DNA does. That's not any different in that regard. All right, so let's back up a little bit. Think all the way back to Unit 2 when we covered macromolecules. We talked about proteins and lipids and carbs and nucleic acids, right? So DNA is a nucleic acid. It's an example of that macromolecule group. Another way to say that is it's a polymer. Remember that word from before. Poly just means many. It's the biggest molecule once you compile all the little parts. We'll talk about the parts in a second. There are two types, right? You have DNA and RNA. And the parts, the little building blocks, the monomers, if you remember that word, are called nucleotides, right? Nothing on this slide is new. It is all recap from earlier, but it is worth going over one more time. And remember that DNA is a double-stranded helix molecule that looks basically like a twisted ladder. All right, let's look at the nucleotide in a little bit more detail. So remember, this is the monomer of DNA. It's also the monomer of RNA. The sugar is deoxyribose. There's a phosphate group, and there's one of 4 nitrogenous bases. So nitrogen bases or nitrogenous bases, it's also right there. So this is the entire nucleotide. All 3 parts must be present in this nucleotide. This purple ball is the phosphate group. And if you're curious, it's not just a phosphorus, it's actually a PO4, and there's a full negative charge on it. You don't have to know that, but just for completeness here. The sugar is drawn as a pentagon because it is a 5-carbon sugar, and each point in this shape is where a carbon atom is, and we'll talk more about that in a second. But that's why it's called a pentose, right? Pent means 5, os just means sugar. So they're saying that this pentose sugar has 5 carbons in it. You guys know that if this is a DNA molecule, then the sugar in question is what? The sugar in question is deoxyribose, right? If this were an RNA molecule, what would the sugar be? The sugar would be ribose, right? And then the last component, again, one of these 4 nitrogenous bases, so adenine, guanine, cytosine, and thymine. So this could either be an A, a T, a G, or a C. And that's it. That's what a nucleotide's made of. So some other details about the nucleotides is that, the A and the G, the adenine and the guanine, are a little bit bigger. If you look at the adenine here, it's a double ring structure. So is the guanine. They're a little bit bigger than the C and the T, right? So the C and the T, if you look here, are actually just single ring structures. You don't have to memorize this at all. It's just kind of an interesting thing to note. The A and the G, with their bigger size, are called purines. And the C and the T, with their smaller size, are called pyrimidines. And the way that I remember this is that like Purina dog chow is like food, and I don't know, maybe you're hungry or your dog is hungry and you want to give it more Purina? I don't know. So the A and the G are purines because they are bigger, they're more. I don't know if that helps or if that didn't, but that's kind of how I think of it. I'm sure there are other ways to go about that. But if you haven't noticed, because what's called base pairing, which we'll get to in a second, remember A's always bond with T's and G's always bond with C's, which means in any pair, one of them is a purine and one of them is a pyrimidine, right? One of them is a purine and one of them is a pyrimidine. So if you have 2 purines, maybe you have an A and a G bonded together, that's a mutation, which we'll get to later this unit, right? All right. These bonds, the A's, the T's, the G's, and the C's, are like the rungs of a ladder. right? So if you had a ladder here, if you drew a ladder, you know, these little rungs, the things you step on might be the G's and the C's, the A's and the T's that are bonded together. And the sides of the ladder are the sugars and phosphates. Right, so we call this the sugar phosphate backbone. So the sides of the ladder, the backbone, are made of these sugar phosphates. Remember, the sugars are the pentagons, and the phosphates are these little balls. Go ahead and label them here. P is for phosphate, S is for sugar. Right, those are the rungs, and then, or sorry, the sides, and then the rungs, the ones that you step on in this ladder, are your bases, your A's, G's, C's, and T's. Let's go ahead and label this on here. P, S, P, S, P, S, P, S, P, and then the same thing on the other side. All right, so here is a little bit more detailed information. It's not a ladder anymore, it's the actual components. So here, again, on one side, the top side, you have your sugar phosphate backbone. Same thing on the bottom side here, your sugar phosphate backbone. And then in the middle, you have your base pairs, C's and G's, A's and T's. That's like the rung of the ladder that you would walk up on and step on, right? So some things to pay attention to here are the bonds. So all of these bonds in between the bases, the T's, A's, G's, and C's. So these dashed lines here that I'm circling right in the middle are hydrogen bonds. These are all hydrogen bonds. They are hydrogen bonds. There are 3 hydrogen bonds, so 3 dashed lines between C and G. Right? Anytime you have a C and a G, you're going to see 3 hydrogen bonds, 3 dashed lines. And there are 2 between T's and A's. Every time you see a T and an A attached, there are only 2 hydrogen bonds there. So technically, T and A are a little bit easier to separate from each other. Technically, not by a lot, but just, you know, just by a little bit. Another thing to notice about the structure of DNA, all of the other bonds are covalent. So all of the bonds, I'm up here on the top, between the sugars and the phosphates, all of these bonds in here, literally any other line that you see in this image is a covalent bond. And remember that covalent bonds are stronger than hydrogen bonds, so they're harder to destroy. And then the other thing you might notice is this 5 prime, this little dash is called a prime, and 3 prime. Think of these as directions, like north or south. There's a directionality to DNA and RNA. And we're gonna look at this here in a second, but if you just do a quick glance, let's look at the top. On this side of the molecule, the right side where my cursor is moving right now, see how the phosphorus, the phosphate group, is kind of sticking out? It's the furthest thing out here. That's always going to be the 5' end of the molecule. If you go to the other end of it, see how the sugar is the furthest thing out? There's nothing past the sugar. The sugar is always the 3' end. And I think I like this. It's easier to remember because the letter 3 looks like, or sorry, the number 3 looks like the letter S to me. So that's an easy way to remember that. And then if you look on the other side, it's the opposite, right? Over here, down in the bottom left, where my cursor is, the phosphate group is the furthest thing out, which means that's the 5' side. And then over here, the sugar is the furthest thing out. There's nothing beyond the sugar, so that means it's the 3' side. And if you look at the numbers, you'll notice that they're opposite, right? The top right, where my cursor is now, 5' opposite of that is a 3 prime. It's not 5 and 5, it's 5 and 3. Same thing on the other side, it's 3 and 5. So this has a name. This kind of orientation, I'm going to write it all the way down here on the bottom, is called anti-parallel. It's called anti-parallel orientation. So let's draw this on the next image here. So I'm just going to simplify it. Here's your two backbones, your sugar phosphate backbones. So I'm not going to draw the nucleotides I'm not going to draw the A's, T's, G's, or C's in the middle. If the tops were both 5 and the bottoms were both 3, that would be parallel orientation. That's not what happens. Cross that off. What actually happens is it's 5 on top and then 3 on top, and then it's flipped. So 3 on the bottom and then 5 on the bottom, right? This is called anti-parallel. That is actually how DNA is organized, right? So where do these 5 and these 3 come from? So let's go ahead and actually draw this out now. I'm going to go ahead and show you. So go ahead and draw a pentagon. Remember, the pentagon is the sugar, S for sugar, and over here, this ball is the phosphate. And just so that we're keeping track of where things are, right here is where your base would be. So you can go ahead and put A, T, G, or C. Or you can just pick one of the 4. Maybe you like A's more than the other ones, I don't know. All right, we're gonna label this. So here we go. At the top of the pentagon, there's actually an oxygen atom. It's not a carbon, it's actually an oxygen atom. And what we're gonna do is we're gonna start numbering here, and we're gonna number clockwise. So on our way to the right, kind of at 3 o'clock, that is a carbon atom. That is our first carbon atom. As we keep going, down at around, 5 o'clock-ish, if this were a clock, this carbon atom is carbon number 2. If you keep going, here's carbon 3. Oops. C3. If you keep going, here is carbon 4. And then the 5th carbon is actually in between that and the phosphate group. So the 5th carbon is actually up here, C5. So this is how we get the numbers, right? So the phosphate group on top is closest to carbon number 5. So we call the side with the phosphate the 5' side. The side down here, opposite of the phosphate, see how the 3rd carbon's kind of all by itself? Like if you were looking down towards the bottom of the page, this number 3 carbon is kind of the one that's exposed. The number 2 carbon's also equally as far down, but it's kind of in the middle of the molecule. This one's kind of out towards the edge. So we pick on the 3 carbon. We call this the 3' side. And that's where the 5 and the 3' come from. They come from the numbered carbons in the sugar molecule, in the deoxyribose sugar. All right, this is just, again, another example of the structure of DNA. Double helix, base pairing in the middle. Dashed lines are your hydrogen bonds. You already know all this stuff. Here's a reminder about your pyrimidines and your purines. Remember, the A's and the G's are your purines. They're a little bit bigger. The T's and the C's are your pyrimidines. They're a little bit smaller. I guess we can go ahead and take note that because the purines are always going to bond to the pyrimidines, the total width in terms of the number of rings of a DNA molecule is 3, right? You get 2 of them from the purines and 1 from your pyrimidines. So your total width of DNA is 3 rings wide, right? 1, 2, 3. 1, 2, 3. All right. So when DNA copies itself, like during synthesis, you have an original strand. So let me back up. So let's say that you unzip DNA so that you're only looking at half of it. And these are the letters you have: A, T, C, G, A, C, G, T, T, A, right? When DNA copies itself, it doesn't put another A, T, C, G, A, C, G, etc., right? It's the matching base. So the matching base for an A would be a T. For a T, it would be an A. You know, you guys know this. This is from middle school, right? C would go with a G. So because the original strand of DNA on top is not the same as what's called the complementary strand that attaches to it, we have to start keeping track of which is the original and which is the complementary, right? But for right now, let's just go ahead and practice. So go ahead, write the complementary strand for this original piece of DNA. You can pause the video if you want. All right, I'm going to assume that you've paused it and gone ahead and completed it. The complementary strand would be T, A, G, C, 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 A, T, T, G, C, A, T, A, G, G. Notice that they are complementary to each other. Complementary. Right. And this sequence is important because ultimately it's going to be in charge of making proteins, which we'll get to later in the unit. All right, that's the structure of DNA. Let's look at the people. Chargaff uncovered the rules of base pairing, which means he figured out that A is bonded to T's and that G is bonded to C's. That's what base pairing is. All right. Franklin took X-ray pictures of DNA using a technique called X-ray crystallography or X-ray diffraction. She actually got cancer from this and died, and then the Nobel Prize that probably should have gone to her went to Watson and Crick, which is kind of a historical component to science that maybe you were familiar with or maybe you weren't. Wilkins was her grad student, so this is actually Franklin here in the picture. But this is the picture that she took, and based off this information, you know, to me and to you, it probably just looks like the letter X. But you know, they know what they're doing, and this actually told them that DNA was coiled. It was actually a coiled helical molecule that Watson and Crick then used to build their physical model of DNA, and they were actually the ones, for better or for worse, that ended up winning the Nobel Prize for it. Notice that it's only 1 year later. This is them, this is their model, and they get credit for building the first double helix model of DNA, the first accurate double helix model of DNA. And then they got the Nobel Prize in 1962. The last little bit here I'm going to go through quick. I'll do this more in a second video. But remember that in order to copy DNA, you have to go through synthesis phase and interphase, right? G1, then S, then G2, right? So all the things that we're going to talk about here happen in synthesis, happen in S phase, right? Remember that if you're a eukaryote, this happens in the nucleus. If you're a prokaryote, this is just going to happen in the cytoplasm. And just for context, one of your cells, or the nucleus in any of your cells, has 3 billion base pairs. 3 billion base pairs. Think of that. That's how many base pairs your cells have to copy when they go through S phase. It takes about 6 hours. So if you want to work backwards, you can probably figure out how many base pairs per second your enzymes have to copy. Oops. All right, when DNA is copied, this is called semi-conservative replication. You do need to know this, semi-conservative. So let me trace the old DNA in my red little marker here. The old DNA, the original DNA, the parent DNA, is called the original strand and the template strand. They're synonyms. You might see either one. Let's actually add parent on there because parent strand is also a term that you might see. So all three of these mean the same thing. So everything here to the left of this point is original strand, right? So let's focus on this one going up to the top. I'm tracing it in red. This is the original strand of DNA that I highlighted in red. Similarly, this other one that goes down to the bottom is also an original strand that I'm tracing in red. Let's do yellow just in case people are red, green. Oh, let's do blue. Bear with me here for a second. All right. The complementary strand is the strand that gets attached to the original strand. So here, I'm doing this in kind of a light blue color, is your complementary strand. It's the strand that's getting put together to complement the original strand. And notice this happens on each side, both the top side and the bottom side. Right? So originally, your DNA was completely red in this case. And here, half of it's now red and half of it's now blue. This is why we call it semi, right? Because semi means half. And conserve means to keep. So we've kept half of the original DNA. If you look at this new DNA molecule, half of it is from the original parent strand, the original strand, and the other half is this blue strand, the complementary strand. This is why we call DNA replication semi-conservative. So here's another image of that. Let me go back to red here. So these were some of the models that they looked at. Conservative replication would have been the original DNA molecule was conserved. It was kept the entire time. So every time it made a copy, you would keep the entire original DNA strand double helix intact. That's not the way it works, right? They were able to eliminate that pretty quickly as an explanation. They looked at dispersive, where like the old parent strands somehow were like in between the newer strands. That also was disproven pretty quick. Semi-conservative is what happens, right? You have the original parents here in dark blue, and in the, I guess you could call them daughter DNAs, but they're not actually called that. Half of that DNA is old, and then the other half, the lighter blue strand, is new, right? And then when those make copies of themselves, it's the same process, right? So by the second generation, you actually have DNA that is completely unoriginal, right? So semi-conservative is how it works. Last bit here, I do know these are a little bit long. I do apologize, but we do have to get through it. The steps of replication. How does DNA copy itself in a little bit more detail? So the first step, number one, is that DNA unzips just like a zipper. Just like a zipper. So an enzyme is going to unwind and unzip the DNA. That enzyme is called helicase, and yes, you must know it. ACE, remember, at the end of a word typically means enzyme. Helix, or hele, tells you that it's unwinding the double helix. So helicase breaks the hydrogen bonds between the base pairs and unzips the DNA. It opens the DNA up just like you would unzip a jacket, right? Step 2 is that a second enzyme called DNA polymerase, DNA because it's going to add A's, T's, G's, and C's, ACE because it's an enzyme, polymer because it's building a polymer, right? You can start to see how the words are made. It's building a DNA polymer, is going to actually make the complementary strand by attaching the nucleotides to it, right? Those are your two main steps. And these are the two main enzymes, helicase and DNA polymerase, that you need to be responsible for. So here's an example. Remember, here is original parent template strand. Remember, there are a bunch of synonyms to describe it. Same thing on the right side here. Here is the original, one of the original strands going up top. Here's another original strand going on the bottom. And it's not shown, but helicase would be right here where this rectangle is. It would be moving to the left here, unzipping the DNA molecule like a zipper. And over here, there would actually be another one. There would be another helicase where I drew that rectangle, and it would be moving to the right, unzipping DNA in that direction. So DNA can have multiple zippers unzipping it, basically, right? You can also have multiple DNA polymerases. So here's your DNA polymerase, this kind of cyan-colored or teal-colored ball. It's attaching nucleotides, making the complementary strand. So here's this new complementary strand right here that's being synthesized or being made by the DNA polymerase, right? We'll talk more about the vocab for this in a different video, but that's kind of the basic gist of how DNA is copied. Last but not least, the why. So why do we talk about this? We talk about this because DNA codes for protein. DNA codes for protein. Organisms are different because of the sequences of their DNA. That's why you're different. If you have an identical twin, they have the same sequence of DNA. Other than that, nothing else on this planet, or probably in the universe, or the multiverse, has the same sequence of DNA that you do. Remember that DNA codes for proteins, and yup, you guessed it, your proteins make up you, right? They're all your enzymes, they're most of your physical structure, and that's why we need DNA. That's why DNA evolved. It is the instructions, if you will, for synthesizing and making proteins. So here's a quick refresher on what proteins do as a throwback to the second unit. They make your body, but I'll let you look over this on your own. Enzymes, hormones, antibodies, none of this is new. This is all recap, and I believe that's it. So I will see you next time.